Hello, everyone. My name is Despo Fata Kasinos. I'm a university professor at the University of Cyprus. And I have the pleasure to welcome you all today in this second morning session, in which we will discuss modeling the AMR threats. And I have the pleasure to introduce to you our keynote speaker, which is Professor Richard Reid-Smith. He's a veterinary epidemiologist at the Public Health Agency of Canada. He is the Surveillance Operations and Targeted Studies Coordinator for the Canadian Integrated Program for Antimicrobial Resistance Surveillance, and he's the co-lead of Risk Work Package of the Genomics Research and Development Initiative Interdepartmental Project on Antimicrobial Resistance. He has co-authored more than 150 peer-reviewed papers on antimicrobial resistance and antimicrobial use in humans, animals, food, and the environment. He is an associate graduate faculty at the Department of Population Medicine at the University of Guelph. Professor Smith, you have the floor. Thank you very much.
Thank you very much for the introduction. So I'm going to describe a modeling approach that we've taken, why we've done it, what it's for, and what its limitations are. For those of you that are engaged in modeling of antimicrobial resistance, depending on what you're trying to model, you'll be aware that it can be relatively straightforward or quite complex. So as said, I'm the co-lead along with Carolee Carson, who is here, and Amrita Bharat, who's not, of the CIPARS, the Canadian Integrated Program for Antimicrobial Resistance Surveillance. I'm also the co-lead of FoodNet Canada with Andrea Nesbitt. So CIPARS is the Canadian program that's equivalent to NARMS that monitors antimicrobial resistance dissemination through the food chain, and FoodNet Canada is more or less equivalent to FoodNet in the United States, which is, at least in Canada, it's a sentinel site-based foodborne disease surveillance program. So CIPARS, I'm not going to go into this in detail, but it is the reason why we do models, primarily in our group anyway. CIPARS has a human component, a diagnostic sick animal component, an on-farm component, a slaughter component, a retail meat component. These are all represented on the top, collecting information on antimicrobial resistance, and on the bottom, antimicrobial use. Both on-farm data from sentinel farms, not across the board, and sales data, as well as some human data. CIPARS is a component of the Canadian Antimicrobial Resistance Surveillance System, which is the umbrella of all the public health agency surveillance systems that deal with antimicrobial resistance. We don't have a water component in CIPARS, but we do in FoodNet Canada, and we are working on sort of regularizing antimicrobial susceptibility testing of isolates from the various sites, the type of water that's collected in each site. We have 4 sites, one in Quebec, one in Ontario, one in Alberta, and one in British Columbia, are different. They're mostly surface water, but we do have some wastewater sample collection in BC. So the part of the issue with our surveillance system, why we do modeling of our surveillance system, for our surveillance system, is to interpret the data that we get out of surveillance in a more comprehensive context. This graphic, which we developed many years ago, modifying the Linton diagram in 1997, is supposed to represent, although it needs updating, the dissemination of antimicrobial resistance through the food chain and the environment. CIPARS really focuses on this component, and most of our models to date have focused on this area, so the transmission from livestock, terrestrial livestock, through slaughter and the food chain to humans. broader scope, we're expanding our modeling efforts to include basically the whole thing to the extent that that's possible. An integrated assessment model, this started out with our original senior epidemiologist analyst, the late Lucy Dutiel, talking to us, our senior risk assessment scientist at the Public Health Agency of Canada, Amir Fazil, about how to look at all of this information together as opposed to in parts. And his idea was to develop an integrated assessment model to try to look at all the pathways together. The other reason that we do it is because policy makers or senior officials often ask what to them are simple questions but are in fact complex questions because they're asking about the whole thing and not just about some specific thing that is amenable to a more targeted model like a QMRA or an agent-based model or a traditional regression model. We've undertaken a number of different modeling approaches, largely associated with the Genomics Research and Development Initiative, but also with some other guises and collaborators. And we've sort of categorized them into ones that are addressing general and specific versus specific questions, and whether they're qualitative or semi-quantitative or quantitative. When we're trying to answer specific questions or look at specific things, we've mostly stuck to the guidance of the Codex Alimentarius. So this with the risk profile followed by a QMRA, and there's several examples of that which will be discussed, I think, in workshop 8 by Dr. Carson. We've also done a qualitative threat assessment that looks at things in general, but when we were trying to do something quantitative in general, we went with this integrated assessment modeling approach. So the confusogram, the sort of diagram that's been shown before and it's shown a lot, looks complex, but the real thing, when you try to pull apart all the bits and pieces and connect all the things that could have something to do with it, especially if they're not necessarily traditional inputs. So this was some of these are diagrams that were the result of a number of graduate students and some different projects using a complex adaptive systems approach. It just, I'm not showing it here to go through the details, but it's just to underline the complexity, and probably a number of you have the same kind of diagrams that emphasize that. So the idea with integrated assessment modeling is that it's supposed to be able to address that kind of complexity, to incorporate data from disparate data systems. It's derived from, originally used in climate change modeling, and can incorporate, in theory, data sets from beyond the kind of things that at least those of us who are microbiologists and veterinary epidemiologists or epidemiologists are used to using. But it doesn't give you absolute answers, it gives you relative answers. It can demonstrate the magnitude and direction of changes in the system in response to changes in the model, but it is particularly appropriate for looking at complex systems. So this was what eventually came to be known as the integrated assessment model for antimicrobial resistance surveillance. Its purpose was to elucidate and quantify the relative contributions of specific agri-food commodities and related exposure pathways to Canadians' overall exposure to AMR from the agri-food production system. So as I said at the outset, we focused our model development particularly just on the food chain. The initial endpoint anyway, was exposure. It's not risk. And it was to look at the number of potential exposures derived from the Canadian population, put in terms of servings at risk, compared to, say, a QMRA. It doesn't require defining a dose or dose-response relationship, but it can account for uncertainty and variability. Each element is developed separately, and then the intent is to try to link them together. So we've concentrated over time on the development of specific pathways or often termed stories. So cephalosporin-resistant salmonella across all food animal species, macrolide-resistant, multiple different kinds of bacteria and broilers, etc. Again, we're looking at the start, we're trying to deal with what's, from a modeling point of view, is kind of the low-hanging fruit in theory, which is the antimicrobial resistant enteric pathogen going on to cause an enteric infection. When we would try to move on to look at commensal bacteria, say E. coli, Enterococcus, Staph aureus, etc., that are essentially commensal or non-pathogenic in the gut, but can go on and cause infection. When they get extraintestinal, it gets and causing an opportunistic infection. Modeling that kind of thing is more complicated, but still, I think it's still the approach to doing it is a bit easier to understand than the bottom part, which is what a lot of the discussion here will be about the environment, the transmission of MGEs and ARGs through the environment, and their potential impact on human health, animal health, or environmental health. Those kind of models are obviously much more complicated. So initially, we're just tackling the top element, although we certainly have aspirations to go beyond that. So the original focus is on the primary food animal commodities in Canada, cattle, beef and dairy, cattle, broiler chickens, pigs, turkeys, and looking at most of these bacteria in each of these things, but not necessarily all of them. So the integrated assessment model is complex in construction, but the math is the model itself is very basic. It's essentially just a series of nodes, and these can be broken into subnodes: farm, abattoir, retail, to the human exposure. And it's really just a combination of risk factors expressed as some kind of measure of association. We've ended up using odds ratios, which I'll talk about in a minute. That factors are either risk factors or protective factors. And those factors can either be modifiable or non-modifiable. The modifiable ones are potentially subject to intervention, or they could be interventions themselves: vaccination, use of probiotics, whatever. The inputs for the model require you to have some idea of, if you give the probiotic, do you get resistant or not? So some kind of conversion into an odds ratio. And those are just multiplied through the model, through each node, starting with an input that's derived either from surveillance or the literature, the input at the farm level. So it's basically a branching probability tree model for each node, and it just knocks on through with the odds ratio, again, dividing and subdividing, and then being summed up back up at the end. You apply that to each node along the way, and then get some estimate of the human exposure. The estimate is the absolute estimate, because there's issues with the estimates that are going to be derived from this. They're not interpretable absolutely. They're interpretable when you put them relative to each other. And then if you have interventions that are in there as factors, you can switch them on or off and look at the relative effect of that. You can introduce new factors. You can introduce new interventions. And in theory, although we haven't got there yet, you can link across the pathways to look at their effect across each other. And some of the background work that we're trying to do is in the environment is aimed at that kind of thing. So here we have a model expansion, a model, at least 3 pathways across species with the same resistance in pigs, poultry, and cattle. And here we'd have a similar thing, but within the same species across different bacteria. This is what one actually looks like. This is the component, just the farm to abattoir section of the 3rd generation cephalosporin-resistant Salmonella Heidelberg pathway in broiler chickens. So these are the On the left are the factors that influence the occurrence on farm, leading to the orange node, which is the probability of occurrence on the farm. Then there's a bunch of slaughter plant factors leading to the slaughter plant on the right, and then that goes on to retail and on to human exposure. The workflow in generating these kind of models, or the kind of inputs to this, is really the collection of, and part of the intent of this is to try to account for the relevant information that's in the literature or that's available through surveillance, or in the case of our work within GRDI from the other work packages. But the primary work is done through literature searches in order to get the information on the risk factors. So those are done through a standardized scoping review process that then identifies the papers that are eligible for factor extraction. The factors obviously have to be extractable in terms of an odds ratio, and that's not always the case, as I'll point out. I'll come back to in a minute. The data are then extracted into a standard database called CEDAR, and then that's inputted into the model itself, which is in a software called Analytica. This is just, a standard literature search string, not really different than doing any other kind of scoping review. The initial work on how we set up the scoping review process is in this publication. As I said, we did settle for the time being on using odds ratios as the multiplier. We tried relative risk, didn't work for a number of reasons. We've considered other measures of association. I think it's something we'll come back to. There's some issues with odds ratios, particularly the model cannot at the moment go beyond Crude's odds ratios. So if we'd like to be able to input adjusted odds ratios from regression models, but it's not set up for that yet. So that's going to lead to inflation, potential inflation or deflation of the probabilities. The information is inputted into the database. This is what the database looks like. So far since we've started, the data from 284 papers have been extracted with 1,272 factors across all the different pathways, leading to 4,690 resistance outcomes. You can see that most of the papers are about Salmonella, with a much smaller number being about E. coli rather, with a much smaller number being about Salmonella, Campylobacter, and Enterococcus. And then the predominant, the papers are mostly chicken and pigs, but there's certainly some cattle papers, and our attempt to create turkey pathways wasn't particularly successful because there's not much information out there. Where the papers come from, we do have some limitations on their applicability to Canadian agriculture, so there's only certain situations where we would take information from outside of Canada or the US, where the agricultural systems are basically tied together, but obviously some factors are going to be relatively universal, and so they can be included in the model. So that looks like not very many papers, but just to look at the Campylobacter broiler chicken pathway, to get 11 factors out of 15 articles, we reviewed 7,344 papers. So that previous slide, to get 284 eligible papers, was many more thousands of papers actually assessed. The decision was to use Analytica, which is a decision modeling tool that's can do probabilistic risk assessments and can handle this model. It does have some issues for us in that when we're trying to deal with our external collaborators, unless they have a license for this, they can't work it outside our firewall, which is a problem because we're trying to do this as a collaborative endeavor. So we have some workarounds for that, but they're not particularly satisfactory. So that's an area that needs further attention. A lot of coordination is required, so using GitHub in order to synchronize everything is pretty essential. Just some examples of some of the pathways. This one is extended spectrum cephalosporin resistant E. coli. So this shows some of the factors. A lot of them are antimicrobial use, but there are other things like litter type, in poultry, conventional versus antibiotic-free management in pigs, conventional feedlots with tylosin ionophore or hormone implants. And there's relatively few that really come in at abattoir or retail. So for example, vacuum packaging was included in the poultry pathway. But we don't tend to get a lot of studies that are done at abattoir or retail that provide the kind of extractable data that we need to create a factor. Fluoroquinolone resistant Campylobacter, at least at the time, this is a bit old, but at the time we did it, there was no relevant data for Canadian beef production. That's actually changed since then, but so there's a lot of different outputs we can come up with. A very simplified version of this in an infographic format. Again, this is fairly old, but this was just looking at the relative contributions of the different commodities with salmonella in red, E. coli in salmonella in green, E. coli in red, and Campylobacter in blue. So it's not really a surprise that chicken would come out higher on all of these things, but it's given that what we did was then to put in a baseline, multiply it through a bunch of risk factors we extracted from the literature. It was at least good to see that it actually gave us what we expected to see, and what if we had just if we'd used our surveillance data to estimate the same thing, we would get a similar result. The lower flag is not the UK; it's actually Ontario. So we did our initial analysis just in Ontario, and then looked at Canada as a whole. So. Where do we go from here? We have a lot of basic stuff that still needs to be done. With the second GRDI project that's been mentioned before, it is more One Health than the initial one, which was based on the food chain. So we're looking at expansion to aquaculture. A lot of the GRDI projects are focused on aquaculture. Inclusion of some environmental pathways. If you look at Poster 403, that's a scoping review that is one of our initial looks at available information. Community pathways, particularly on ESBLs, because if we're trying to interpret the contribution of the food chain towards resistance in human health, we have to see how it intersects with what's going on in the community. So this is moving on to this lower area, both commensals becoming opportunistic infections and hopefully the dissemination of the impact of ARG or MGE dissemination. We also, as described this morning by Irina, want to move beyond exposure to human health impact, including burden of illness. We really should be able to construct the same sort of nodal propagation beyond exposure. We have some issues with temporality that need to be addressed. We need to relook at the odds ratio, as I mentioned. And also, we haven't got very far in one of the intents with the model, which is comparing, controlling the risk factors, removing them, introducing new risk factors to see what sort of relative impact that has. That's just describing what I mentioned about adding the human health impact. And then a key thing, obviously, is how do we improve the model with genomics? Most of the inputs, especially the risk factor inputs, are based on phenotypic studies that use phenotypic methods. Do those translate to genotypic results, or if we have genotypic results now that are better at lumping and splitting the bacteria as they move through the food chain, the ones that tend to move on and the ones that tend to get held back, whereas phenotypically they all look the same. And so then the odds ratios were calculated on a group at the different points on groups that aren't necessarily the same populations, even though they might look like that phenotypically. That application is not restricted to the integrated assessment model. Obviously, it also applies to any other of our, modeling or risk approaches, including risk profile, and that will be, I think, discussed in workshop 8. There are a number of other risk workshops where I think a lot of these issues are going to come up as well. We have ideas about application and incorporation of genomic data, but haven't set on anything in particular. One of our grad students has been, who's the technical lead for the model, has been playing around with beta diversity. Not sure that's the answer. So basically, in summary, the integrated assessment model, well, that's a lot of work, but we did a lot of pathways. So each pathway is actually relatively faster to build than if we were doing a QMRA or even a risk profile. A lot of the outputs of what we've been doing can feed the other modeling approaches because they tend to, we are doing scoping reviews on the same thing. But when you fit all this stuff together, it doesn't give you an absolute estimate, it only gives you relative estimates, but the factor effect modification can be assessed. There's a lot of diversity between serotypes on the effect of factors. I think one of the universal findings, which isn't going to be a surprise to anybody, is that there actually isn't enough data about the specific things that we're looking for. So almost invariably, one of the key findings is we couldn't fill this part of the node, or we couldn't find a risk factor for this node in the propagation because there's no study that actually shows it, either because it's not been done or because it wasn't reported in a way that we could actually extract the data. And as I mentioned, there is this issue of, for example, with Analytica, that if we're trying to be open and collaborative about this, we have some limitations. A lot of students and a lot of people have worked on this over the years. It looks like a lot of people, and it is, but a lot of them are Masters of Public Health practicum students who are working on 4-month stints. So we've had a number of funders over the years, including Genome Quebec funding Dr. Thomason's component, and a number of associated projects, and I'll stop there. This is not about the models, it's about our surveillance reports, but if you're interested, that's where you can find them.
Thank you for your presentation. I'm Jing Huang from Tsinghua University, and my question is, when you think about assessing the risk of AMR for the foodborne pathogen, it looks like you just focus one pathogens, right? But in the environment, I want to know that how we can assess the risk of AMR, because there are diverse bacteria in the environment.
It's a good question. I mean, and it's not even really correct for foodborne pathogens. We just pick the ones that are the big ones, but there's plenty of others, and plenty of things that could be transmitting. So we've picked, like everybody else, indicator bacteria. So you could do that in the environment, but if what you're really interested in is the movement of mobile genetic elements, or ARGs, then that's probably what the focus should be, and I think we don't know how to do that yet, and maybe that will come out in some of these workshop discussions.
Thank you.
I think there's maybe a couple of presentations that are getting at the start of it, so we'll see what comes next.
Thank you very much.
So there's a question there, and then Professor Tom Tsang.
Nicholas Ashbolt, University of South Australia, over here. My question is in relation to the literature you're using. It was unclear to me whether you're gleaning prevalence data for antimicrobial resistance versus rates of exchange of data.
It's the risk factor. So it's essentially if you have a factor and resistance or not resistance. So vaccination, yes, no, resistance, no resistance. Or probiotics, yes, no, resistance, no resistance. So if it can be expressed or as an odds ratio or a risk factor or relative risk, we can use it. If we can't, then we can't use it. It doesn't mean the paper is wrong or invaluable, it's just we can't extract data from it. And that's a discussion that we've had in GRDI. It's often the question about how can we package our data so that, like the work package data, so that we can extract it. And in some cases, the study is just not set up that way, and that's not a flaw in the study, it's just we can't use that for our model. That's just a reflection of the simplicity of this model, and obviously in other kinds of modeling, there's other kinds of data that you can use, and going back to the beginning and the potential inputs of the other kinds of data, that really hasn't been addressed yet, and it should be.
And you mentioned something about the dynamic nature. Are you going to include sort of a temporal aspect in future editions?
We're certainly trying, but there's more than one issue with temporality. Like the basic one is the timing of some of the risk factors versus what comes, particularly when you have like one animal that is slaughtered around 30 days and one animal that is slaughtered at over a year, and when they're exposed to antimicrobials, and when the food comes out the other end, you know, they're not all the same. So the cycles are quite different.
Thank you.
And that's not really well dealt with.
Hi, Tong Zhang from the University of Hong Kong. My question is about integration of metagenomic data or genomic data into the model. For your opinion, I think, what could be your opinion on about the relative abundance of antibiotic-resistant genes and pathogen or MGE, or the absolute abundance of this kind of different components? What could be the role?
I don't know yet. I mean, we don't know yet. I mean, it's probably going to be relative abundance, but I don't know. Like, go to workshop 8. I think we'll talk about it there. We really don't know, and that's why we need EDAR.
Hello, everyone. Yeah, it's my great pleasure to be here as the co-chair for this session. Actually, I'm Bing Li from Tsinghua University, China. I'm very lucky because I got my visa in the last day before my flight, so I can act as the chairman for this session. So next, we will invite Dr. Hunter Kwon from Arizona State University to give us a talk. So, Dr. Kwon, please.
Hello, everyone. Okay, so today I'm going to present an assessment of risks associated with antibiotic-resistant E. coli. Just a little bit about me. I'm a postdoc right now at Arizona State University, and before that I was at Oregon State and University of California, Irvine. That's just a little outline of my presentation, and I'll just go ahead and go right into it. So, as we're all well aware of, many of these presentations have suggested that wastewater treatment and wastewater treatment plants have been identified as a ARB and ARG hotspot through selective pressures and horizontal gene transfers. But I'm very excited to finally move over here to the after part. You know, how does this deal with exposure and health risks? What about after treatment? How can we quantify and better understand those risks? The two main portions of my talk today focus on the quantitative microbial risk assessment framework, shown here if you're not familiar with it. And QMRA is a framework used to assess the risk of a health outcome such as illness, infection, or even death from a given exposure to an identified pathogen of concern. It's based on data and modeling methods such as Monte Carlo simulation, which is a technique used to predict the probability of an outcome, like infection, as I said, based on randomness of the different input variables to each of the parts of the framework shown in the figure on the right. So for my part one, it is a risk-based assessment of antibiotic-resistant E. coli in reclaimed wastewater irrigation of lettuce. Identifying some of those main categories of the QMRA. We start with the hazard identification, which, as I said, is identifying the pathogen of concern. And for this portion of the talk, I'm basically doing a demonstration of a new type of a model for quantifying the risks of infection for AMR. So I sort of went the easy route, and I chose E. coli as my identified hazard, as mentioned, because fecal coliforms are regulated in wastewater reuse applications and for its high prevalence both in the environment and for the frequency of possible exposures, especially through agriculture for consumers such as you or the farm workers themselves. So given that type of an exposure, I modeled the consumption of lettuce that is irrigated with reclaimed wastewater. And I don't show any of the big equations here, but I'm happy to talk about that afterward if you'd like. And then quantifying the exposure dose, we move into the dose response assessment, which is the mathematical relationship between the exposure dose and the probability of the chosen response. So whether that's an infection, a major illness, or even death. And what makes this study unique is that we expand upon this traditional forward framework to try and account for the antibiotic-resistant bacteria within a given dose. So I'm going to focus here on the dose response assessment portion, which is the most unique for this study. And just briefly mention that if you're not maybe familiar with QMRA as much, the two models that are implemented and fitted to dose response clinical data the most are the exponential model, shown here, where D is the dose and P is the probability of the outcome. And the exponential parameter R is essentially in a survival probability, which assumes that each organism or each of the bacteria in a given dose has an independent and identical survival probability, essentially in the body, and that there is a constancy of the pathogen host survival probability. The other model is the Beta-Poisson model, shown here, where similar assumptions, however, this R, this survival probability, in this case, comes from a beta distribution, which is sized or fitted based on the alpha and beta parameter. A few years ago, Chandrasekaran and Zhang developed what they called a simple death dose response model, which I'm going to talk about a little bit here. And it's based on those previous models that I talked about, but also taking a step back and being based on the birth-death process, or in this case, the simple death process. And the birth-death process or simulation is a form of a continuous time Markov chain, shown in this picture where I believe one of the earlier speakers today talked about it. And essentially, between each time step over a simulation process, you have a random transition from one state to another. So if you start with 0 bacteria and then you move forward one time step, the simulation may predict that a bacteria will be born, increasing the total number by 1. And then you move on, so forth, based on given or identified birth and death rates, so that pathogens may increase by 1 or decrease by 1 with each time step. The simple death process is that same idea, but we're only going to include the death rate. So if you start with a pathogen dose, let's just say there's 100 or 100 people or bacteria, whatever that population is, over time, there's going to be some rate of death. So every time step, they either maybe stay the same or decrease by 1. So that's the fundamental idea of what this dose response model is based upon. And shown here on the picture on the left is just a little graphic showing that. And it follows the idea that if you're exposed to a given dose, over some amount of time, that entire dose will experience total death and nothing will happen to you. There will be no response. Or at some time threshold, if the dose is large enough, there is some probability that they will regrowth and subsequently cause an infection. So the simple death dose response model combines that fundamental idea with the previous models that I showed. For example, shown here in the equation is the simple death dose response model version, I like to think of it, of the exponential model, where we take the survival probability of the exponential model, R, and relate it to this simple death rate mu, multiplied by the time point that I show in the middle of the graphic, TFS, raised to the power of the total dose. So the larger the dose, the larger the probability of survival. In addition, if we're talking about antibiotic-resistant bacteria, there may be the chance that these death rates will be different depending on whether they are resistant or susceptible to antibiotics. So if the dose is exposed to any level of antibiotics, we include them in the model. Therefore, we separate the dose into two subpopulations, resistant and susceptible, and are able to estimate the probability or the death rate for each one separately. So like I said, I wanted to take this model and apply it to a QMRA approach with data that we're used to in the microbial risk assessment world, especially when we're looking at a possible pathway, which is consumption of irrigated lettuce. So I'm going ahead and just showing you the results of this simulation where I took that model and I took an exposure model that I developed for this consumption of irrigated lettuce, and then I sort of just played around with all of the other parameters. So on the left side, I'm varying the concentration of the E. coli in the reclaimed water that's being used to irrigate the lettuce, and then I vary the fraction or the fraction of the dose that is either resistant or susceptible to antibiotics. And on the right side, I held all of those values constant, and I instead changed the value of trace antibiotics that the dose would be exposed to. Some of the key takeaways is we see that on the left side, if you have a higher fraction of ARB going from 0 to 0.4 or 40%, the line goes from black to red, and it goes up by maybe about half an order of magnitude or close to 1 order of magnitude. Therefore, a higher fraction of ARB in this case produced a higher relative risk. If we go to the right side, we sort of see the opposite effect. The higher the C, or the residual antibiotic concentration, the lower the risk. We have the black line, the black solid line, and then going down we have the dotted red line for the increased antibiotic concentration. However, in both cases, we see that that dotted line is red. So in this case, based on the probabilities of the population survival, we predict both the probability of risk and the probability of the infection being caused by either the resistant or the susceptible population. So if it's caused by the resistant population, we're calling it an AB, untreatable infection, which is the red line. So in both cases here, if you increase the fraction of resistant bacteria or increase the C, you're more likely to cause an untreatable infection. So real quick, I'm sure you're all thinking, why should I care about this? You just played with a bunch of numbers. There's no real data there. Well, I have a surprise for you. I did the same thing, but I had a very lucky partnership with a team in Spain where they took some data for 3 greenhouses where they used reclaimed wastewater to irrigate tomatoes. And I did a very similar exposure model. This time, instead of lettuce, it's tomatoes. So some of the exposure parameters are a little bit different in there. But I used real E. coli data, which included both the total E. coli and the resistant CFUs for each sample. And I did some similar results. So for this, we see that for the first greenhouse, the risk was pretty much non-existent. Most of their samples were below detection. But we see the same trends in greenhouses 2 and 3, where as you increase the antibiotic concentration C, the line goes from black to red. In other words, more of the resistant bacteria will not survive, leaving the resistant bacteria to cause the infection. And yeah, I'll also just say that overall the risk was very small, but we saw the same trends in resistance. And briefly, I just want to say what's next? Where can these models go? Well, I made this picture on the right just to demonstrate that a lot of samples, especially with reclaimed wastewater, are taken after treatment. They're taken after the treatment plant for regulation purposes. But not just with E. coli, but with all these other pathogens that we could be considering, there may be room for modeling or for predicting how these resistant fractions may change either over time or all the way at the end to the point of use. And I briefly talk about a couple models here that are used both for dose response and for bacteria simulation. And I just want to briefly say that this was another demonstration I did where I included HGT, or horizontal gene transfer, in a simple death model. basically just to show that only on the right side, where if we increase the HDT rate by several orders of magnitude, did it play a role. So I just want to say that there is room for all of these other resistant parameters, such as gene transfer, to be included in dose-response models, but a lack of data is currently hindering that, I would say. I'll skip my takeaways and next steps because I believe I covered all of them and I'm out of time. And I just want to briefly thank my current and previous mentors, Carrie Hamilton from Arizona State University and Sunny Zhang from University of California, Irvine. Thank you.
Mary Shane, Solar Environmental, thanks for the awesome talk. I'm wondering what you think of fitting your simple death models to the kinetic data instead of the human dose response feeding study data to create a new dose response relationship.
What kind of kinetic data?
So if you had kinetic data of feeding studies in mice of a really interesting resistant pathogen of concern, or I don't know what they call them because I'm not a lab scientist, but when you make like a mock organ and you model the kinetics of a particular pathogen in that simulated environment.
Yeah, I would say that's a great question because I'll say actively right now that's kind of what I'm trying to work on. I'm trying to take that very last picture I showed with all the red and blue curves and try to do a, maybe not just a simple death version of the simulation, but a birth-death simulation and integrate it with a risk assessment so that maybe if you have growth above a certain threshold, then you would have a risk outcome occur. But so far, I haven't spent that much time looking as I haven't had the data. So I would say, yeah, if you have an in vivo kinetic model or kinetic data for bacteria over time after a given dose, then you could fit those birth and death parameters or rates to that and then do the risk after that. So that's what I would like to do.
Okay, one short question.
I hope it's short. Heike Schmidt from the Netherlands. I was wondering, the dose response curve you're using is for STEC, right? It's
yes.
So it's an infectious dose response curve. Did you consider, or do you have any idea how to perform risk assessment for colonization by a non-infectious E. coli strain? I know it's really difficult, but just to hear whether you have any ideas.
So the risk of colonization, that's what you would want the outcome to be?
Yes, because often it's not direct infection that follows, but it's colonization of your intestines. And then over time, maybe if you're hospitalized, then you would develop infection, right? So I think that could be kind of an intermediate step in your dose response models.
Good point. I think kind of like with the kinetics question, there's definitely room for that if you I would say take a step back from the traditional dose response and return to those last points I was talking about with more of a simulation approach or a bacteria interaction type model where you put in population limited growth or birth and death, and then, like you're saying, predict how the numbers change over time based on exposure. I don't immediately have an idea of how to translate that yet to response or to what you're saying of the risk outcome, but I think that is a good next step. Yeah. Thank you.
Okay, next speaker, Professor John Woolridge-Kraft from the University of Birmingham. Professor Kraft is a computer scientist from the computational biology. Please.
Thank you for the nice introduction, and thank you all for coming, and thank the organizers for the opportunity to speak. So I will be talking about AMR Flows project, which is a collaboration between Indian and UK scientists. All the data is about the Indian environment. I've promised too much in the abstract in the title. I'm not able to cover all of that in 12 minutes. Apologies for that. But I do want to give you an overview of the whole project. We wanted to do a lot of measurements and mesocosm studies and remote sensing, and so to feed into models of antimicrobial resistance dynamics and models of water flow, and combining them and feeding that into the quantitative risk assessment. So we'll only talk about some of the field measurements today in the quantitative risk assessment. We have been doing some of the other work, but due to lack of funding, we have not managed to do all the work that we planned to do because we got paid 2 years later than we needed the money. So some of the things are just now starting since we got some more money now. This is the Musi watershed, central in the Musi where most of the sampling points are. Sorry, I can't point at them. Is Hyderabad city, a big city, a small river, and we have one sampling point upstream of the city and 2 sampling points downstream of the city. I hope you can see it, I can't point at them. And they're quite far downstream from the city. There are about 25 point sources within the city that we know something about, some quantitative information. There may be more that we don't know about. And the basic pattern is along the river that you start with low concentrations upstream. Dry season is red dots. Then it jumps up in the city and it drops down downstream of the city. In the wet season, the change is more gradual. Dissolved oxygen, opposite picture, so high upstream, dropping down to 0 within the city and slowly recovering downstream. Wet season again a bit more mixed. 16S, also like nitrogen, jumps up in the city, drops down downstream, not quite as far than it was upstream. And one example, resistance gene, has this pattern that it jumps up in the city and drops down downstream, but not to the level it was upstream. Another type of pattern is CTXM, where it drops down to the same level as it was upstream. So these are the 2 types of pattern we got with resistance genes. So what these data suggested to us was that the river can be divided into 3 stretches, upstream, within the city, and downstream. And doing clustering, you can see this pattern very clearly. Upstream and downstream cluster together, very distinct from the city samples in the dry season. In the wet season, a bit more mixed due to the more gradual increase. The downstream sites and the early city sites are clustering together now, whereas the second part of the city is still on its own, and upstream is far away from that. So I've done lots of permanovas to show that all of these is that the stretches and the seasons are significantly different for almost all the variables we've observed in the water column. In the sediment column, it's a bit more complicated picture, but still a large number of variables are significantly different in terms of stretch and season. So I can throw hundreds of p-values at you, death by p-value. Okay, so what we've then asked is whether we can explain the pattern of resistance genes using other data, in this case water quality measurements, to predict or to discriminate the resistance gene, the stretches that we found by clustering resistance genes. So we are using a separate data set to explain the resistance gene pattern. And here you can see with linear discriminant analysis that you have a clear separation between the city locations and upstream. Upstream is furthest away, hope you can see it on the right, top right, and downstream is in between, which I think is a sensible pattern. This is just showing 2 variables here, dissolved oxygen, total nitrogen. We have more than that, and it works a bit better if you use more than 2 variables, but the 2 I think are sufficient. I don't have time to show more, but there is a poster on that by Vika Sonka. On Thursday, I will present it because he couldn't get a visa to come here, so it will be me talking about the poster if you have more questions about the Musi data. So moving over to Chennai, we wanted to use this river to use the river as a comparison to the Musi, because this doesn't have industrial pharmaceutical input, but is otherwise similar, small river, large city. Now the problem with Chennai was we didn't find any clean enough upstream sampling sites to have a contrast. So we tried again earlier this year to find some upstream sites that are cleaner. We did find them, but they are not connected to the river. We have sampling points. The first two ones, TNR and PR, are outside the main city, but they are already quite polluted. And then we have the sampling sites within the city, and then downstream we have to stay far enough from the sea to avoid tidal influence. Okay, so here are some example results. Water quality, often it's fairly flat. There is no change along the river, but there are some exceptions. It's quite a complicated pattern, and we haven't fully understood it. I've asked the team to find out why we have certain jumps at the last two sampling sites, and they've done a lot of work, and we still don't understand what's going on there, but we hope to find the reasons for that. What we have done this year is to find the cleaner upstream sites. They are not connected with a line to the other data because they're not from the river. And you can see low nutrient levels, nitrogen, carbon, phosphorus, and high oxygen levels, pretty much the same 16S abundance. Looking at resistance genes along the river, you can see that, so the first one is 16S, the second one is the E. coli marker, and the rest is resistance genes, that the sampling locations in the river tend to be higher in resistance levels, but sometimes it's not much difference really to the cleaner upstream samples. There's still a poster up there from Tara that you can have another look at when you're interested in the Aria River, and I'm also trying to answer questions to that. So now moving to the risk modeling, we've decided to use a Bayesian network for that. When I started with this project, I had read some papers from Rupert's group using this method for risk assessment, and I thought this is a good way to do it. So we are now using this in this project as well. It's a probabilistic graphical model. More technically, it's a directed acyclic graph, and what that means is there is no feedback. That's the main disadvantage of the framework, that there is no feedback. The core of it is that you have nodes which have a conditional probability table. That's the heart of it. So we can do probabilistic inference forwards and backwards, which is good. This is a simple example. If you have a population with low levels of COVID, and you have the antigen test and the PCR test, given that the antigen test has a higher false positive and false negative rate, you have a higher positive detection level in that case with the antigen test and the PCR test. So this is basically conditional probabilities. Right, so in our model we cover wastewater, river, crop irrigation, and exposure. I don't have time to go into all of these modules, but I'm only going to show the exposure module at the end. So we follow the source pathway receptor pathway with these modules. And in each module, we are interested in the concentration, so absolute abundance of resistant bacteria, sensitive bacteria, and antibiotics. So, thank goodness Hunter just explained to you the model by Chandrasekaran and Jiang, so I don't need to go into that. It saves me some time. So the endpoint we're interested in is if someone becomes infected or not, and if he or she becomes infected, whether that infection is treatable or not. So that's the endpoint that we're interested in where we're using this model. Basically, if you're starting with exposure, there are different types of exposure that lead to ingesting a certain number, given the amount of water ingested, you end up with a dose. The dose then feeds into probability of being resistant, of the number of resistant bacteria in that dose, feeding into a probability of extinction of the resistant bacterium in the body or no extinction. Good, 1 minute left. So that gives me the probability of finishing this on time of 90% or something. We also include the antibiotic in there. It's all feeding into the next node with these condition probability tables. So what I wanted to explain very briefly is that we can also output from a mathematical model into this system. I'm going to demonstrate this very briefly using this relatively simple ordinary differential equation model of resistance with sensitive resistant bacteria and antibiotics, which you can simulate, and anything else you can simulate you could use. So you have to do lots of simulations with all the different conditions, different parameter sets. You end up with 50,000 or so simulations, which gives you results which you have to bin or discretize into small number of categories, otherwise it becomes untractable. And then you can learn conditional probabilities in the Bayesian network, and that gives you these outcomes. So what I wanted to demonstrate is that you can use expert information, you can use measurements, you can use an analytical mathematical model. That's easy, you can just write it down, or simulation of a model as input into the Bayesian network. So it's a very flexible approach. So to summarize, Musi River pollution is driven by sewage, which masks the pollution by industry. Aria River is a less clear pattern, but the seasonal pattern is different to the Musi, and you have just seen what the Bayesian networks look like and why we like them, and thank you very much for your attention.
Actually, I have the same question as was asked before. Shouldn't it be possible with this model also to model the probability of colonization, and shouldn't that be even easier?
I think everything is possible with the Bayesian network approach. You can model the probability of colonization. We have kind of lumped this together with the probability of extinction in the body because we're using this simple death process model. But you could also model the probability of colonization, which might be, if you're interested in, someone travels somewhere and picks up some resistant bacteria while traveling, and then the probability of colonization, which then maybe only later on leads to infection or transfer of the resistance gene. This can be done, but we haven't done it.
Okay, thank you.
Nicholas Ashbolt. Yep. Nicholas Ashbolt, University of South Australia. You mentioned the acyclic nature of the Bayesian networks that you're working with, but of course you could use dynamic Bayes nets to get around that issue for example, looking at the build-up in sediments of antibiotic-resistant genes and so forth. So have you considered that sort of dynamic approach to deal with that issue?
We have considered that, but given how far we are and the complexities of what we already have, we haven't attempted that. What you can of course do is you can include any kind of feedback in a mathematical model that you then feed into the Bayesian network. So in that way you can integrate feedbacks, but there is no easy way to do it directly. I was told, I'm not the expert in the Bayesian network models, that is not my own work, but that's what I was told, that it's not so easy to do it. You would have to iterate through the model many times.
Come to workshop 9, thank you.
Yeah.
Mary Shane, Solar Environmental. I'm wondering if you were to point to one part of your model that was most important and needed more data, what would you point to?
I think there are many parts where it's not very clear between what's in the river and human exposure. I think that area is least clear. We have been actually trying to use zebrafish as a model of human exposure. That kind of doesn't work because they don't get infected so far, so we don't have that input. But the idea was that the fish swim around in the water and swallow the water, and you can get a relative risk of infection by resistant or non-resistant bacteria, but that hasn't worked yet.
Thank you, Dr. Lee, for the introduction. Hello, everyone. I'm a PhD student in the Computer Science Department of Virginia Tech. My research specialization is metagenomics and computational biology. Today, I'm going to talk about a bioinformatics tool that we designed and developed to compare metagenomic samples in terms of risk associated with antibiotic resistance genes. Antibiotic resistance genes anchor cellular functions that enable bacteria to survive in the presence of antibiotics. ARGs are ubiquitous in the environment, but not all ARGs present the same level of threat to human health. In 2015, Jose Martinez and his peers proposed a conceptual framework called risk to determine the potential health risk of environment originated from ARGs. In this framework, residents were ranked based on the criteria like the mobility of ARGs, whether they are being hosted by pathogens, the novelty of ARGs, and the corresponding antibiotics the ARGs are resistant to. This framework is applicable on metagenomic sequencing data and can be utilized to determine which environments are more concerned to us. Such information can be very helpful in environmental policy in making decisions like resource distribution or mitigation efforts. The concept of resistance risk has been utilized in designing several computation pipelines in the next few years. For example, MetaCompare-1 was published in 2018. This pipeline takes assembled contigs as an input data and outputs a number called resistance risk score. The risk score was calculated based on the ARGs found in the sample, associated mobile genetic elements, and the human pathogens that are hosting the ARGs or MGs in the sample. The pipeline was applied on 14 publicly available metagenomic data, and it was shown that the samples collected from locations that are highly contaminated has higher risk scores. In 2021, a framework called ARG Ranker was published. This framework divides nearly 4,000 ARGs into 4 ranking groups based on 3 criteria: anthropogenic enrichment, whether the ARGs are being carried by MGs, and whether they are being hosted by escape pathogens. In 2022, another group published another framework that uses similar criteria but some additional refinement of their own, like clinical availability of the antibiotics that ARGs are resistant to. Both of these frameworks rank ARGs based on the known associations with MGs and pathogens, and that's their key difference with MetaCompare, because MetaCompare ranks metagenomic samples based on the existing associations that are found in the sample. But MetaCompare-1 has some limitations. For example, the pipeline used in its pipeline needed to be updated. It uses acclaimed database for MG annotation, and this database contains accessory ARG sequences along with MG sequences, and that results in false positive annotation of ARG as MG. It uses Patrick database for pathogen annotation, and Patrick contains information of only 12 pathogens. Secondly, in calculating risk score, MetaCompare does not distinguish which ARGs or pathogens are of more concern to human health. And finally, the risk score is not very intuitive. It can range from 17.53 to infinity in theory. We designed MetaCompare 2.0 to address these concerns. In this tool, we replaced the previous database with 3 state-of-the-art databases to annotate ERG, MG, and pathogens. We redesigned the risk score calculation so that it's now more intuitive and can range from 0 to 100. We introduced 2 distinct risk scores that we call them as ecological resistance risk and human health resistance risk. In calculating ecological resistance risk, we are using all the ERGs observed in the sample, their associated MGs and pathogens. Here we are trying to estimate the overall potential of ERGs to mobilize into pathogens. On the other hand, the human health resistance score is calculated based on a selective set of ERGs and pathogens that are of acute concern to human health. To validate our tool, we mined literature to find samples that represent surface water, wastewater, soil, sediment, and gut microbiome. For each of these environments, we obtained 10 samples that are collected from locations which are highly contaminated and 10 samples from locations that are less contaminated in comparison. We broadly subcategorized these 2 contrasting groups as polluted versus unpolluted. And when we compared the risk score, we observed that for all environments and for both resistance risk score, the polluted samples always have higher risk score, and the difference was statistically significant. I have told before that the input to MetaCompare 1 is assembled contigs, and that's also the same case for MetaCompare 2.0. Now, assemblers can vary in accuracy, and their performance can depend on sample coverage and complexity. To prove the robustness of our pipeline, we used 5 samples and assembled them using 3 different assembly methods. When we finally compared their risk score, we noticed that the risk score of a particular sample can be different based on what assembly method you are using, but that their rank order that remained the same irrespective of the method you have applied. Sequencing depth can also affect the coverage of the assembled contigs. Now, to check what would be the impact of sequencing depth on the resistance risk score, we performed a simulation study. In this study, we used 3 wastewater samples collected from effluent activated sludge of a wastewater treatment plant. And the original samples, they were very large and deeply sequenced, and they contain nearly 4 billion short reads. And from these samples, we generated subsamples using random subsampling method, and those samples have different range of sequencing depth. They can be very shallow, like containing only 1 million short reads, or very deeply sequenced, containing like 250 million short reads. So when we compared their risk score, we noticed that the sample that simulates shallow sequencing, they have their risk score can vary, and the rank order can be highly different. But the samples that have coverage higher than 60%, then their risk score is kind of stable. So in our previous examples, we are comparing samples that came from different locations, but they are kind of from the same environment. Here we are running another study to compare samples across multiple different environments. We are basically trying to see what would be the range of the risk score in all of these environments. So again, we mined literature to collect samples from 11 different environments. So for each of this environment, we obtained nearly 100 samples. And when we compared the risk score, we noticed that the influenza samples have the highest ecological rhizome risk, and the chicken feces samples have the highest human health rhizome risk. And the results were as we expected. So all of our test results are shown as based on short-read data. Theoretically, you can apply this tool on long-read data, but we are yet to test it on long-read data. So our next plan is to test on long-read data and make necessary adaptation. And also in our next iteration, we plan to refine the risk score based on the different categories of mobile genetic elements, and also incorporate clinical importance of antimicrobials. So this is all I have, and here I would like to thank my co-authors who have helped me in data collection, in designing these experiments, and also I'd like to thank my mentors for their continuous guidance.
Thanks for your talk and for developing these interesting tools. My name is Zhiguo Zhang from Westlake University, China. And my question is that, what do you think about the results of meta-compare is comparable between different samples, like between wastewater and soils? Because this environment, their microbial complexity is greatly different.
Yeah, I know. So your question is, I did not properly understand what you are exactly asking. So you're saying that we cannot compare samples from other environments that are
from different environments.
So here I'm just I'm trying to show that what would be the situation when you apply the meta compare in these environments. What would be the expected risk score you might get here?